In this video, we'll talk about fluorescence recovery after photo bleaching or FRAP. This video is in English version. If you need a Hindi version of the same video, you can click on the I button or in the description. So let's learn the concept of FRAP in next 10 minutes. We'll learn about the principles of FRAP, how FRAP is performed, applications of FRAP, and limitations of this technique. So fluorescence recovery after photobleaching is a technique to study molecular dynamics. Let us quickly review the applications of FRAP. So it can be used to study membrane fluidity. It can be used to study mobility of membrane proteins, can be used to study cytoskeletal dynamics, and to investigate intracellular transport like vesicle transports. All of these are some applications of FRAP. So let's quickly go through the analytics part. This chapter is in CSIR Unit 13. The questions that are asked, generally they are in part B or part C. I'll give you some example at the end of the video. The difficulty level is overall easy if you understand the concepts behind FRAP. So the key things that are asked is basically the application and the principle, which is on which the FRAP is based on. So the resources that can be used is the classical Pathfinder books that every CSIR net aspirant use. So this particular chapter is in the biophysics and molecular biology book, or in cell biology, that means the life science part 1 book. You can get it in both the places. Anyway, one can also use Wilson and Walker principle of techniques and all of these things. So I won't recommend Wilson and Walker if you don't have that much of time. You can also refer to Molecular Biology of the Cell by Bruce Alberts. That is the ideal reference, but again, it depends on your time. Anyway, hit that subscribe button before we start the video. So let's understand the principle of FRAP using a biological context. So the biological context is how membrane fluidity can be experimentally determined, right? So we are thinking of an experiment that we have to determine the membrane fluidity. And in order to ask this question, we can utilize the technique called FRAP. Before we begin, we have to understand the setup that we require for FRAP. We need a confocal microscopy setup, and that is generally set to a live imaging mode. That means the microscope has to be covered, the temperature has to be controlled like this. So entire microscope is kept in a box like this, which has attachment for other mobility of gas and liquids that we need for our cells to stay alive. So this is a live imaging confocal setup, and we can perform FRAP using this. Now in FRAP, you need lasers. So obviously you need a confocal microscopy setup. It can't be exactly done using fluorescence microscope. So here is a cell, and we want to look at the membrane fluidity of this cell. How we can do that? We can fluorescently label these membrane using a fluorescent version of lipid. So if we wait for some time and incubate these cell with this particular lipid construct, it would incorporate that in the membrane. And now our sample is ready for examination in FRAP. So now we are looking at the membrane from the top view, and this is the side view. First we would be illuminating so we would get a measurement of the overall fluorescence of this sample. Then we would illuminate with a very high power laser that would bleach the fluorescence in that particular region. So small patch of membrane would be exposed with very high intensity laser beam. As expected, that particular portion will be bleached as shown here. Now what we do, we wait for some time. So this bleached out membrane should be recovering over time if there is a molecular dynamics. So membrane is fluid, so other particularly fluorescently labeled fluid or fluorescently labeled phospholipids would come from the adjacent region and try to fulfill this bleached region. So in the bleached region, we should be seeing some amount of fluorescence recovery. So after photobleaching, there should be a recovery if there is a molecular dynamics. If there is no dynamics, no movement, then recovery would not be happening. So this thing can be understood in format of a graph. So let us try to understand this graph to interpret the data. So this is how the normal baseline fluorescence look like, and this is when we are imaging the overall membrane patch. We haven't exposed the high intensity laser right now. Now we'll be putting the high intensity laser. So whenever we put the high intensity laser, it would bleach a portion of the membrane, that is expected. So what is meant by photo bleaching? Your fluorescence intensity at that particular region would go dramatically down. Exactly in the graph you can see the intensity, which you can see in the y-axis, has gone down quickly, sharply, right? And this is somewhat visualized like this particular cartoon. And then if you wait for some time, if there is dynamics, fluidity, then there should be some recovery. So you look for the recovery profile. So this is really important. In FRAP, in order to understand molecular dynamics, one bleach the specimen and then look for the recovery kinetics. The recovery kinetics tells us a lot about biological processes and dynamics. So let us try to understand what recovery profile tells us. So let's study membrane fluidity using FRAP with some live example. So before that, we'll talk about what can govern membrane fluidity, such as temperature can govern membrane fluidity. Higher the temperature, more the kinetic energy. So there would be overall more fluidity in the membrane, right? Also, lipid composition can govern membrane fluidity. If you have more kinks in the double bond in the phospholipids, then possibly the membrane fluidity would be better. So unsaturated fatty acid can make the membrane more fluid compared to saturated fatty acids, right? And also cholesterol composition can widely affect membrane fluidity. More the cholesterol, it works like a dynamic glue, so it would prevent membrane fluidity, increase membrane rigidity. In this situation, we can see there are three particular membrane and they have different membrane composition. The first one has a lot of unsaturated fatty acids. The second one is entirely composed of saturated fatty acids, and the third one is a mixture of saturated and unsaturated fatty acid with some amount of cholesterol in it. So obviously, if we look at we want to understand the membrane fluidity, which has the highest membrane fluidity, we can look at the FRAP profiles, right? The first profile, which is in red, correspond to the membrane which has unsaturated fatty acid. Look how the recovery profile is pretty fast. Now the particular one which has only saturated fatty acid, it has an intermediate recovery profile shown by these green or bluish green traces. Look at the purple one, it has high level of cholesterol. That means it makes the membrane highly rigid. So the recovery is first of all delayed and the recovery is also partial. So just by looking at the recovery profile, even if we don't know what is the membrane composition, in retrospect we can understand or we can assume that this particular membrane might have a different lipid composition that alters the fluidity. Let's study vesicle dynamics using FRAP. So one scientist can ask a question like how neurotransmitter containing vesicles are reaching the synapse from the cell body. So obviously there are several highways which are composed of microtubules, and on the top of these highways the trucks such as kinesin would take the vesicle from one side to another side. A scientist can possibly label these vesicles with a fluorescently labeled protein or some other means. So we need to label these vesicles by some means, and we can ask questions that whether they are dynamic, in which direction they are moving. So we can look at the movement time scale, movement kinetics, and movement direction. So in this case, what scientists do, they bleach a portion of the membrane in the neuron. They perform FRAP, obviously. And then they look again the recovery profile over time. And the recovery profile would give us an idea about how dynamic the vesicles are. It can give us an idea that what is the time scale of this movement, what is the movement kinetics, and overall, to some extent, it might also give you an idea about movement direction. So now we understood the principles of FRAP and its application with some live examples. Now let's look at the exam corner to understand what are the previous year questions. So in CSIR NET June 2016, they asked a question that lateral diffusion of protein in membranes can be followed and diffusion rate can be calculated by atomic force microscopy, scanning electron microscopy, transmission electron microscopy, or FRAP. So if you learn the principle of FRAP properly, you can clearly understand this particular problem can be understood by FRAP. So you bleach a portion of the membrane and try to understand how this particular portion is recovered by dynamics from nearby molecules. If the nearby molecules diffuse into this particular region, you can understand the membrane fluidity is high, and also you can monitor this over time to calculate the recovery profile and the diffusion rate. So the FRAP is the correct answer. Question number 2 is for you guys. This is in CSIR December 2016, and this is in part C. They say fluorescence recovery after photo bleaching is a method to estimate diffusion of a molecule in the membrane. Fluorescently labeled molecules such as a receptor tagged with green fluorescent protein, a receptor labeled with GFP which interacts with cytoskeletal, a labeled lipid, a labeled protein that binds to the membrane surface are photo bleached. So there are 3-4 molecules which are labeled with GFP. Now they are going to be photo bleached and looked for the recovery profiles. Question is, there are some pseudo recovery profiles given here labeled as A, B, C, D. You have to identify that which recovery profile correspond to what and whose time scale can be somewhat comparable. So this is the question. So again, it is based on the application of FRAP and basic understanding of FRAP. If you can solve this question, put it in the comment. Okay, again, if we quickly look at a part B type question. So this question is not asked in NET, but it's just a sample question. A cell biologist was studying the kinetics of our intracellular vesicle trafficking by tagging a cargo with fluorescent labels. Which of the following technique is best for him to quantify the dynamic? So first of all, fluorescence recovery after photo bleaching, FRAP, phase contrast microscopy, scanning electron microscopy. So obviously you can understand in order to look for the dynamics, the scientist can use FRAP, right? So I hope this concept is clear right now. We have looked at some live examples, we looked at some questions, but if you need more questions, you have to subscribe to this channel and stay tuned for more updates and more question answer videos. So you can get a list of question practicing video in the I button and description very soon. It's not uploaded yet, but it will be soon. Anyway, don't forget to like, share and subscribe. Please follow this channel, share this channel with your friends, and let me know in the comment whether these videos are useful or not. If you have a suggestion, put it in the comment.